Hello, welcome to another bioinformatics video. In this video, I'll be talking about hierarchy, distance metrics, and phylogeny. But first, some review. How do we fetch column 2 from this NumPy array? So in this case, since it's a NumPy array, we can use the comma-based row-column indexing system. We can see that it's a multi-dimensional NumPy array. So basically a matrix. If we were to print this out, we would see a rectangle that is 3, 6, 7 elements long and 2 elements deep, or 2 elements high. So we can use this approach here, and using the order of rows first and then columns to index correctly. If we want column 2, then that basically means we want 0, 1, 2, this number and this number, the 2 sevens, right? Because these are the rows, this is a row, and a column would be cutting through those rows. So we can use this format, all rows, comma, column 2. So this would be all rows of column 2. So we want both row 0 and row 1, fetching the position 2 in each case. So C is the answer. How do we fetch the 999? So here we're going to see there's a 999 in position 0, 1, 2, 3 of, so this would be column 3 of row 1, 0, 1. And let's see, if we look at this, we have all rows of column 3. So that would be the 4 here and the 999. And then of that result, we can fetch element 1 of that. So since this would be element 0 and this would be element 1, that would give us the 999. If we take a look at this option here, this would fetch us first row 1, and then on top of that fetch element 3 from whatever is produced by that. So if we fetch row 1, it's going to be this one, and then element 3 of that would be 0, 1, 2, 3. So that also gives us the 999 in this case. Option C, we have the NumPy row column indexing notation here. So row 1, column 3, so that's the most direct. Row 1, column 3. So all 3 of these are correct, so the answer is D, all of the above. How do we change each value to a 1? And here we have my array equals numpy.zeros. So it's a 16 element long list of zeros, array of zeros. And we have 3 for loop related options here. So the first is going to loop through the elements in my array. So I will take on the values of this zeros array. So on the first round through, it would be a 0. The second round through, it would also be a 0, and so on until it goes to the end. So it'll be 0, 0, 0, 0 the whole way. And this operation down here will be changing the iterator into a 1 for just that round. And then it will go back, and then I would change back into the next element, like another 0. And then we would say I equals 1 again. But actually, this I is not directly connected to the data here, right? So this the data here is being used as the source for creating I. So you can think of I as, in some sense, a copy of the elements inside here. So changing I is not actually going to affect the source material here. So changing I is only going to have the effect of changing I for that iteration. And then it's not going to actually do anything to the original array. So A is not correct. For I in range 0 to length of my array of item I equals 1. So this is the correct one. Here we're looping through a range 0, 1, 2, 3 up to 16, and then we have we're going into my array at the respective position, so position 0 or position 1 or position 2 and so on. And in each of those cases, we're taking whatever is stored there and setting it equal to 1. So this then would give us the correct answer here. If we want to change each value in this array to a 1, change it from a 0 to a 1, this would be the way to do it. Option C, for I in range 0 to length of my array, I plus equals 1. So this here starts the same. We're again going to have an I that's going from 0, 1, 2, 3, 4 up to the end of the length of this array here. And then here we're doing something similarly wrong as A, right? We're just changing the iterator, and the iterator is not really connected to anything. And in this case, this notation is indicating that we're trying to increment it, add 1, but it's also being set to 0 each time. This will have the exact same result as this, basically just make I equal to 1 for one round only for it to be immediately set to 0 in the next round, having no effect on anything. So B was the answer. And then, so a comment about the course. If you were new to the course or new to programming, now you're part of the coding community. So I encourage you to continue doing programming. It's a useful skill, and you can continue to build on it and expand your knowledge. And there's a large community, especially with Python, that you can contribute to. There are many forums, websites like Stack Overflow or the Bioinformatics Stack Exchange are completely maintained by its community, by users, professional and amateur coders alike. There's also GitHub, so this is like a sharing platform where you can collaborate with other coders and upload your own material and share it with the world. Another recommendation is something called Rosalind for bioinformatics, and this has the same philosophy that we've been pursuing in the exercises, which is that you can learn this material by trying to do it yourself. So Rosalind is a set of problems basically related to bioinformatics, and some of them are harder than others. You can see distribution of different, how often people get the problems right when they submit. And a lot of people are using Python to solve these problems. And some of these problems have been even the inspiration for exercises in this course. For example, things like making functions that translate DNA to DNA and so forth. But the outline for the next couple videos will be to cover these topics. So we'll be discussing hierarchy in nature and phylogenetics and applications of trees and hierarchy to multiple sequence alignment. So starting with the first topic, what is hierarchy? Hierarchy is, first of all, a mathematical structure. It indicates relationships, and objects in a hierarchy often occupy a rank or a position. And groups of objects can be ranked as well. So this is a dominant feature of hierarchy, that you have groupings. The structure is recursive, so the same rules apply to superstructures as to substructures. And that will become more clear as we go through this. But here's an example of a hierarchical structure. This is a tree diagram, and this is a type of network diagram, specifically one that is composed of, so it does not have cycles, for example. So in a tree, at least in a formal tree, you do not have a situation, for instance, where this node here can reconnect to another branch, right? You only have sprouting events, nodes sprouting initially from a root and then branches out to finally leaves on the end here. So trees have these objects that form the joints in the structure, and these are called nodes or vertices usually. And the outer ones are usually referred to as leaves, and the first one is referred to as the root. And we have connections, which are links between these, and these are usually referred to as edges or links. So here's a simple example of an encyclopedia and how it's organized. So we could organize it into different sections: the culture, the art, craft, the science section like this, where art and craft are subcategories under culture. Then we have another representation format, which is the enclosure diagram. So for example, we could have these structures enclosed and further structures within that, and structures within that. Oops. And this represents the same information. We have substructures and ranks, but in this case, it's indicated by what is enclosed by what. But this essentially is the same type of information that's being presented in the tree. And here you can see we have the exact analog of the tree here with this encyclopedia example where art and craft are contained inside the culture box here. In nature, hierarchy is extremely common. So first of all, there are social hierarchies. So for example, in ants, we have a hierarchy, a social hierarchy, where you have maybe many workers and a single female queen that yeah, the ant society is organized in a kind of pyramid. We also have things like food webs where you have just a couple, say, great white sharks for every Chinook salmon we might run into, and for every one of these salmon, even fewer, even more numerous slimy sculpins. And somewhere at the very bottom of this pyramid, we have primary producers, algae and things that are extremely numerous. Similarly, with wolves, we have these social structures. You also have tree structures, hierarchical structures occurring in other places in nature. Of course, trees, regular trees, obviously have something like a tree structure, and snowflakes also have kind of branching patterns that form from a single root or origin. The distribution networks for plants and blood vessels and lungs, these distribution networks have a tree-like structure. Things like rivers and fjords, these also have branching tree-like patterns that emerge. So both living and non-living things tend to exhibit this pattern. In artificial systems, we have lots of hierarchy. So for instance, the encyclopedia here and how it's organized. File systems on computers are hierarchical. The structure of computers, how computers are organized, and the memory, for example, has a hierarchy. Literally every piece of technology. So cars, for example, cars are composed of subcomponents, large modules, right? And these modules form the major functional parts and body and the kind of final assembly. But those parts themselves are composed of subparts, and then those subparts are composed of still more subparts, the nuts and bolts at the very bottom level. And if you were to assemble a car, it wouldn't make sense to start from the nuts and bolts to start assembling the entire car just from the base components. You would go module by module, assembling subcomponents before you fit everything together. Language also has structure. So sentences themselves are hierarchical, right? You can break them into clauses, and you can break clauses into objects and actions, and you can break those phrases into still smaller divisions. The families of language languages and where they're derived from. Like English and German, for instance, are more related to each other than English and Spanish, for example, because they share a more recent ancestor than English and Spanish do. So this language, all languages have this kind of hierarchical pattern as well. So just some food for thought, and I'm not going to answer it because I don't know the answer, but something interesting to consider is, why do hierarchical structures emerge in nature or in artificial situations? And are there different types or categories that separate the various examples? Are there general classes? And do all naturally and artificially occurring hierarchies share something fundamental in common besides their tree structure? So yeah, just some food for thought. And then we'll move on to a really important tree, which is the category of phylogenetic trees. The most obvious example being the tree of life itself. Right, we have large branches like bacteria, archaea, and eukaryotes. There are a couple ways to present these trees. So you can have rooted trees where we're explicitly showing the positions of ancestors, and then unrooted phylogenetic trees where we're just showing the relative relatedness between present day groups. And of course, many of you are probably familiar with this taxonomic ranking system where we have, for example, the animal kingdom at the very bottom of this pyramid, and then the small individual species at the top. And yeah, some terminology here. We have taxon, a population or group of populations of organisms, which are usually inferred to be phylogenetically related and which have characters in common, but which differentiate the unit. For example, geographic population, a genus, a family, an order, from other such units. So phylogenetics, it's the study of evolutionary relationships. And we have here an example of this with Charles Darwin's notes here. And you can see he was, back in that time, was thinking about these relationships. And this, of course, is striking because this was before bioinformatics existed and before the structure of DNA was known. And he was able to deduce these relationships based on morphological characteristics, like the shape of beaks of these finches. And today we have modern methods. So this is a paper from not too long ago going into Darwin's finches and creating a very high resolution phylogenetic tree of how they're all related. And in contrast to the morphological approach of Darwin's era, we have today molecular approaches for phylogenetic analysis. And this is usually based on nucleic acid or protein sequence data. Hierarchical rank can mean different things. So there are different ways to draw these trees. So you have, for instance, the cladogram, which this axis, the length of these connections here, it has no meaning, right? The only thing being presented here are the connection data, the topological information, but there's no actual metric on this scale here. In contrast to that, we have the phylogram, which is explicitly showing some kind of genetic change on this axis. There are more mutations between this point and this point, for example. And then correlated with genetic change, but not necessarily completely proportional to, we have time. And this is another way you can organize these trees. And sometimes, for example, with the ongoing coronavirus pandemic, you have a situation where you have both temporal and genetic information that can be applied to constructing these trees. So these trees, what determines their structure? What is driving them? Well, it's natural selection, survival of the fittest, right? So you have, for example, a trait here, and some change occurs in this trait. So we might call this trait A, and then if we follow this particular lineage here, we see that trait A has changed into trait A star. And for whatever reason, this trait is more fit, and we get a larger number of progeny coming out of this lineage than we do out of the original trait A lineage. And then this is going to drive the structure of these trees. This is going to be the major driving force in determining which branches die off and which branches continue. It's really the generator for these shapes. So how do these traits actually change? So we have molecular evolution through substitutions, first of all. So we have mutation by nucleotide substitutions. These have a particular frequency, and these frequencies are not all the same. They can depend on the genetics of the organism. They can depend on environmental influence. For example, animals or plants living in the shadow of the Chernobyl accident. They have a strong environmental factor influencing their mutation rate. Position in the genome can matter. Genes versus non-coding regions, right? This has a big impact on how deleterious, for example, a mutation might be if it affects the function of a gene versus perhaps a less important non-coding region. Or maybe some of these non-coding regions are important. Maybe they're regulatory functions, and you could have deleterious effects there as well. Particular sites in the gene. So some areas are more mutable than others. If you have a protein that has a binding site, maybe that binding site will be very sensitive to changes, whereas other kind of structural bulk in the protein is more mutable. Also, which gene? Not all proteins are as critical for preserving life or promoting proliferation. Fixation, how much evolutionary pressure there is due to natural selection. So there are neutral mutations that become fixed at a base mutation rate. You have things like synonymous substitutions that don't change the protein, and these end up getting used to estimate mutation rates. Then you have non-neutral mutations, and these tend to either rise to prominence in a population, like we have over here, or are quickly snuffed out. And here we can see variable mutation rates in bamboo here. So you can see this particular species is changing faster than these ones. And in humans, we have numbers like this, 64 mutations per generation. Also, the mutation rate of male sperm is higher than the female egg. And then mitochondrial DNA, for example, undergoes more mutations per generation than genomic DNA, for example. We have things like PSSMs, position-specific score matrices, that can be used to represent evolutionary constraints. So if you remember, we have columns here that correspond to positions in a peptide or a protein, and amino acids here listed as rows. And the values in these matrices represent log odds probabilities of these particular substitution events relative to the base mutation rate, and they differ by position. So, for example, this the F in this position here is many orders of magnitude more favored than the other options in this column. And so the result of that is going to be there's a strong evolutionary pressure to maintain an F here. If a mutation to something else occurs, we're going to see a strong likelihood to return to the F here. In contrast to that, rows like this one, we have a smaller evolutionary preference for one particular amino acid in this column. Instead, we've got 3 here that have the same order of magnitude score. And this means we're more likely to get fluctuation, for example, between these 3 most likely candidates here. The rates of evolution in different proteins. So fibrinopeptides, responsible for coagulation. These peptides have a high tolerance for mutations because their only job is basically to be present when clotting happens. They don't have a lock and key function in the same way that enzymes have, for instance. The histone has extremely low rate of change. These have a super precise geometry and a critical survival function, right, for organizing chromatin. So mutations in histones are not tolerated. The mutation rates in things that are not undergoing strong selective pressure changes, these things that are mutating or varying at a constant rate, these can be used as a reference for time. So this is called the molecular clock hypothesis. And the idea is basically that for a mutation rate of 100 mutations, for example, per, per million years, this basically says that if you have 2 species that have 100 differences between them, then from that we infer that there was a common ancestor approximately 1 million years ago. And so here we can see if we plot the rate of change here, of amino acid differences, it should be a straight line. It should be a linear relationship if we're going to use it as a molecular clock. And that way, the rate of change is proportional to time. And then we know, therefore, that if we see a certain number of changes, this corresponds to, roughly corresponds to a certain time. And this is how we can link phylograms and ultrametric trees, the information in those with each other by using molecular clocks. And then the time frame we're interested in matters as well, right? So mitochondrial DNA is changing at a faster rate than other types of DNA. So if we're interested in a very short time range, we might use mitochondrial DNA, but we should use it in the linear regime. So if we try to extrapolate further with mitochondrial DNA, we have problems with it becoming a non-linear relationship with time. There's a number of challenges to this molecular clock hypothesis. So the main idea is that mutation rates are not constant. They tend to fluctuate, at least, and they can change with things like generation times. So, for example, my generation tends to have children at a later age than my grandparents' generation, for example. So this can skew the perception of time if you're looking at data that's varying only by generation. Population size and genetic drift can be smaller in small populations, so more mutations are neutral. Species to species differences, things like metabolism, ecology, and evolutionary history can influence mutation rates. And things like changing selective pressures, things like climate change, for example, can distort the molecular clock. So statistical models then get used to correct for these issues. But the thing to keep in mind is that while the goal is always to predict these rates, models are not perfect, and this is always a kind of approximation in the absence of more precise temporal data, data such as that which we have for coronavirus, for example. Other ways genes can change. So besides substitutions, we have things like increases in genome size. We humans are diploid organisms. We have 2 copies of each gene, but there are tetraploid organisms out there. You can also have things like regional duplication. So this can happen internally in genes. For instance, the immunoglobulin protein family tends to be, so this is IgG, for example, and it is composed of these subunits here that are derived from duplication events. You have whole gene duplications that give rise to different proteins. So the IgG family, for example, has many members that share a common ancestor, but they are today separate molecules, all residing in a single system in every person. We have IgM, for instance, involved in the early immune response, and IgG, the classic antibody, involved in the later adaptive immune response. So yeah, the effect of these gene duplications can be to extend the physiological repertoire, like with like immunoglobulins, for example, or also things like the globin family, myoglobin, alpha, hemoglobin, all derived from a single common ancestor, but serving different roles. And the gene duplications can result in changes in function. So lysozyme, originally a bacteriolytic defensive agent, but then became adapted to serve as a digestive function. So some terms, we have, of course, homology, just the similarity shared due to relatedness. They share a common ancestor. Orthologs, so these are homologous genes that are descendant from the same ancestral sequence, but separated by a speciation event. So mouse and human IgG, for instance. Paralogs, this is like human IgA and human IgG, right? Within the same species, but diverging from within. And then finally, analogs, and this is convergent evolution, right? If you have something that's derived from a different ancestor but converges on the same function, and this can happen, for example, with histones here and this bacterial HNS protein that do something very similar to each other. So that's it for this video. I'll see you in the next one, where we will go into more details in how to construct trees. Thank you.